Hello everyone, grab your pen and paper and let's talk about type 4 hypersensitivity. In this lecture, we're going to be understanding the general mechanism and a brief overview of the type 4 hypersensitivity. Essentially, the T helper cells, the CD4, and the cytotoxic T cells, the CD8, are going to be responsible for tissue damages and inflammations. Also, phagocytic cells could be involved in this type of hypersensitivity. Now, it is worth noting that it is also called a delayed type hypersensitivity due to its relative, there's a brief delay between the effect of this type of hypersensitivity in contrast to the other types. Now, following an infection, an antigen-presenting cell, which could be, well, plasma cells, they could be even dendritic cells, for example, they're going to be presenting antigens on their MHC class 2 to T helper cells. And generally speaking, these are T helper 1 cells because there is a release of the interleukin 12, which is going to be promoting its effect, or its proliferation, essentially. Now, next, upon activation, the T helper cell is going to be releasing cytokines, most notably the interleukin 2. And this interleukin 2 is crucial for the proliferation and the production of other T cells, and also inflammatory cytokines, namely the interleukin 1, the TNF alpha, and the interleukin 6. Now, at the end, the cytotoxic T cells, the CD8, also play a major role in this type of hypersensitivity as it contributes to the release of certain cytokines. But in the context of the type 4 hypersensitivity, it is going to be recognizing antigens that are exposed on the MHC class 1. Recall that the MHC class 1 is present on all nucleated cells. So they're going to be presented on the MHC class 1 of normal cells, and they are going to be inducing damages by the effect of the release of the perforins and the granzymes. And the perforins, from their name, they're going to be forming pores, just like the MAC, which is the membrane attack complex from the complement system. It is also going to be leading to ionic imbalances and also going to be leading to the bursting of the cells. Next, we've got the granzymes, which are going to be inducing apoptosis, which is the induced cell regulated cell death. Now, the pathogenesis of type 4 hypersensitivity, it is a result of the breakdown of self-tolerance mechanisms. Now, I'd like to address this a little bit because pretty much every type of hypersensitivity that we have mentioned there is a role of the central tolerance or self-tolerance that is involved. Because if there wasn't a problem by heredity or other problems that are related in our body, these types of hypersensitivities will not occur in the first place. Now, in short, inflammation causes an influx of self-reactive lymphocytes to our tissues. This is relating to the first type of the first part, the one detailing the CD4 cells that are going to be causing these inflammations in the first place. So these inflammations are going to be causing an influx of self-reactive lymphocytes to the tissues. Now, these are self-reactive due to reasons mentioned before, obviously. These self-reactive lymphocytes are going to be inducing damages in normal tissues present in our body. So let's conclude the two different ways the cells might cause problems in this type of hypersensitivity. First of all, we've got, as we see in the image to the left, we're going to be seeing the effect of the CD4 and the CD8. The CD8 is going to be recognizing the exposed antigens that are present on the MHC class 1, and this is going to be inducing damages in normal tissues. And recall that they have to be nucleated ones because the MHC class 1 is only and exclusively present on nucleated cells. Next, we've got the CD4, and then upon activation of these CD4, they're going to be releasing certain cytokines, and these cytokines are going to be leading to the inflammations, which are also going to be damaging to our cells. Now, this concludes our overview of type 4 hypersensitivity. I hope you learned something new. This has been Ali from B Biology.